In this lesson, we'll be talking about acid-fast staining. So acid-fast staining is another differential stain. So with a differential stain, we use two or more dyes to stain different types of cells or different parts of a cell. In an acid-fast stain, our intent is to differentiate between what we call acid-fast positive bacteria and acid-fast negative bacteria. Some bacteria have what we call a waxy cell wall. These bacteria are typically within the genus Mycobacterium. So having a waxy cell wall is a unique characteristic to Mycobacterium. Most other bacteria will lack having a waxy cell wall, and for that reason will stain blue, which is negative during an acid-fast stain. However, with Mycobacterium, such as Mycobacterium tuberculosis, this unique characteristic of them having a waxy cell wall helps them to hold on to the primary stain in the acid-fast stain, which is carbonyl fusion, and in the end they will stain pink. So in this differential staining technique, pink is positive, and it means the bacteria have a waxy cell wall, whereas blue is negative, which indicates the bacteria do not have a waxy cell wall. Like I said, the only really genus of bacteria that produces positive results with an acid-fast stain are Mycobacterium, and for that reason, acid-fast staining is a diagnostic tool that can be used for tuberculosis patients. So if someone has active TB, you can collect a mucus sample from them and do an acid-fast stain on that sample. If it comes back pink, which is positive, you know right away they're infected with a Mycobacterium, and then based on their symptoms, you would probably assume Mycobacterium tuberculosis. Now, if it came back blue, that's negative. In that case, they probably have pneumonia caused by some other bacteria like Streptococcus pneumoniae. So a little bit more about this waxy cell wall. Like I said, only Mycobacterium will have this waxy cell wall, and the reason why they have a waxy cell wall is because of these substances called mycolic acids that are embedded within the cell wall. So this is what gives them that kind of waxy texture. And these mycolic acids, and the fact that Mycobacterium have a waxy cell wall, it's actually a protective structure for them. So one of our body's main defenses to protect us from TB is to use phagocytic cells. So phagocytic cells in general will engulf bacteria and then release digestive enzymes to ultimately destroy those bacteria. The exception to that role is Mycobacterium tuberculosis. When macrophages engulf the TB bacteria and they release the digestive enzymes, unfortunately the TB bacteria is protected from those digestive enzymes by the waxy cell wall with the mycolic acids. So it actually lets Mycobacterium tuberculosis survive in kind of a dormant state within the macrophages. And then at some point, if the individual becomes immunocompromised, the bacteria can be released and then cause an active infection within the lungs. So it's actually a kind of virulence factor that Mycobacterium tuberculosis have. But when we do an acid-fast stain, if the bacteria have this mycolic acid and waxy cell wall, that allows them to hold on to the primary stain when rinsed with alcohol. So that's what allows them to stain pink in the end. So to kind of go through these steps, when we do an acid-fast stain, the first thing that you would have to do is your standard smear prep that we've learned in other lessons. But once you're ready to start staining, the first thing you would do is you would start out with the primary stain, which is carbonyl fusion. So initially, before staining, nothing has color, they're just transparent. So we use the primary stain carbonyl fusion, which typically is going to sit on the slide for 5 minutes, and this typically is done using steamed so that the waxy cell walls can kind of really absorb the stain if they're acid-fast positive. But initially, when we stain with carbonyl fusion, whether the bacteria are acid-fast positive or negative, they all stain pink because that's the only stain that we've used. Then you'll rinse with water, and then the next step is to do the decolorization step, which with acid-fast staining, you have to decolorize with acid alcohol. We use the same technique where we do one drop at a time on the slide while holding it at an angle so we can watch the drops run until they turn transparent. But when we rinse with the decolorizer with the acid alcohol, if the bacteria are acid-fast positive, they will stay pink. Their waxy cell walls allow them to hold on to the carbofusion pink dye so that they can remain that primary color. However, acid-fast negative bacteria do not have the waxy cell wall, so when you rinse them with the acid alcohol, they get stripped of this carbofusion color and are temporarily colorless. Then we would rinse with water, and the very last step is we have to stain anything that lost its color during the decolorization step. So we apply methylene blue to the slide. That typically sits for a minute or two, and that's going to stain any of those acid-fast negative bacteria blue color. So in the end, once all is said and done, if your sample is pink, that means you have an acid-fast positive bacteria that has a waxy cell wall with mycolic acid. You're likely looking at some type of mycobacterium, like Mycobacterium tuberculosis. If your acid-fast stain produces blue results, that's a negative reaction. Your bacteria lacks a waxy cell wall with mycolic acids. And while we can't definitively say what bacteria you're looking at because there's millions of bacteria that are acid-fast negative, you at least know you're not looking at Mycobacterium.